Good morning. It's February 26th, and this is your daily brief in science. Here's everything you need to know. A small set of pRNAs in blood may predict 2-year survival in adults 71 and older with up to 86% accuracy, outperforming age, cholesterol, activity, and more than 180 other indicators. Lower levels of particular pRNAs were consistently linked to longer survival, suggesting these molecules may influence aging and longevity. Higher pRNA levels could signal underlying health issues. The strongest signal comes from a combination of a few pIRNAs, pointing to a practical, minimally invasive blood test for short-term survival risk. The findings were published February 25, 2026 in Aging Cell and funded by NIH institutes and related aging translational science initiatives. Results were validated in a separate, independent cohort of older adults, reinforcing robustness of the pRNA survival signal. The observed pRNA pattern mirrors findings in model organisms like C. elegans, where reduced pRNA levels are associated with longer lifespans. Future work will assess whether lifestyle changes or medications such as GLP-1 drugs can modify pRNA levels, and how blood pRNA levels compare to tissue levels to clarify mechanism. Plans include exploring therapeutic avenues to modulate pRNA levels and testing GLP-1 receptor agonists with attention to systemic versus tissue-specific pRNA dynamics. Lead author Virginia Byers-Krause highlights the potential for a practical blood test to identify short-term survival risk and guide strategies for healthy aging. The study uses AI and machine learning to analyze 187 clinical factors and 828 small RNA types from blood of over 1,200 participants, with validation in an independent cohort, underscoring the robustness of the pRNA signature. Compared with lifestyle and conventional clinical measures, pRNA markers provide superior short-term survival prediction and could enable risk stratification, personalized care, and timely interventions in geriatrics. Across broader horizons, pIRNAs outperformed traditional risk factors for short-term survival prediction, while for longer-term survival, lifestyle factors gained influence, but pIRNAs still offer valuable added insight. A team of UNSW researchers has unveiled a process to produce high-quality graphene from discarded peanut shells, aiming to cut costs and reduce environmental impact. The method starts with lignin-rich peanut shells, pre-treated to form a carbon-rich char around 500 degrees Celsius, then uses flash joule heating to briefly reach about 3,000 degrees Celsius to transform into graphene. Under Professor Guanyo, preheating creates char before rapid flash joule heating for mere milliseconds to yield single-layer graphene at approximately 3,000 degrees Celsius. Graphene's strength, conductivity, and transparency underscore its value across electronics, energy storage, medical devices, and flexible tech, highlighting scalable, low-cost production. The work appears in Chemical Engineering Journal Advances, with authors including De Cacchino, Cordero IM, Lin B., Gia M., and others in 2026. Commercialization is anticipated within 3 to 4 years, with potential to adapt the method to other lignin-rich wastes like coffee grounds or banana peels. Researchers are exploring other organic wastes as precursors, citing lignin content as the common factor enabling similar graphene production. Peanut shell graphene could transform agricultural waste into valuable materials, enabling large-scale biomass to graphene manufacturing for electronics, energy storage, and flexible technologies. The process can produce graphene in roughly 10 minutes, with energy costs estimated around $1.30 per kilogram, suggesting competitive production economics. Overall, a kilogram of graphene using this method is projected to cost about $1.30 in energy, with the entire workflow completed in about 10 minutes. Initial work identifies lignin-rich peanut shells as the key precursor, with pre-treatment to remove impurities essential for achieving high-quality, defect-free graphene. This approach uses no added chemicals and relies solely on peanut shell-derived char, reducing energy use and environmental impact compared with traditional graphene production. A new learning framework blends incremental learning with transfer learning to predict energy use in directed energy deposition for metal additive manufacturing, improving accuracy with limited data. Published in NPJ Advanced Manufacturing, the approach supports robust, real-time predictions and cross-scenario generalization across different materials and process conditions. Across tasks, models that combine incremental learning and transfer learning outperform vanilla models, with LSTM and TCN producing the strongest results and transformers showing gains in certain cases. The workflow follows 3 stages: source domain incremental pre-training, target domain retraining, and target domain testing, with explicit parameter updates through incremental learning. Material transfer across alloys causes a larger domain shift than transferring laser power within the same material, but incremental learning narrows that performance gap. The neural network uses backpropagation, dropout, and batch normalization. Memory buffers and meta-learning help retain knowledge, while domain-adaptive transfer learning aligns features to minimize distribution differences. The study uses data from 20 samples of Kokermo or IN 718 to conduct 3 transfer tasks, transferring knowledge across materials and processing conditions. While promising, the work notes limits in generalizability under substantial material or process changes and suggests future directions like GAN-based data augmentation and physics-informed simulations to enrich training data. Future work aims to expand to more materials and processes and to integrate energy forecasts with mechanical properties and surface quality for a holistic optimization platform. Diverse directed energy deposition datasets, including variables such as laser power, scan speed, and powder feed rate, contributed to notable improvements in prediction accuracy and resilience to catastrophic forgetting via memory replay buffers and meta-learning. The research highlights environmental and workforce benefits, emphasizing energy-efficient manufacturing and the augmentation of human expertise with predictive analytics. Incremental learning enables continuous model updates as new data arrive, while transfer learning supports knowledge transfer across related directed energy deposition scenarios to reduce labeled data needs and speed learning. A citizen science find off Cairns has revealed what may be one of the largest Pavona clavis colonies documented, estimated at 111 meters long and about 3,973 square meters in area. The discovery was made by Sophie Kalkowski-Pope and her mother Jan Pope, who used measurements, video, and a 3-dimensional model to confirm the size. Researchers then applied spatial modeling and high-resolution imagery to verify the dimensions and create a 3-dimensional representation for future one-to-one comparisons. This 3-dimensional mapping supports ongoing monitoring to track changes over time and assess reef recovery in targeted areas. The exact location remains undisclosed to protect the site from disturbance, in line with conservation best practices. Experts say such 3-dimensional mapping and systematic monitoring are essential to inform protection strategies for vulnerable parts of the reef. The find occurs amid global and regional coral bleaching tied to rising ocean temperatures, highlighting the importance of reef monitoring and protection efforts. The colony's appearance resembles a rolling underwater meadow, and it could be a single ancient coral or multiple coalesced colonies. Confirming origin would require extensive genetic sampling. Conditions at the site include strong tidal currents and relatively low exposure to cyclone waves, and scientists are exploring whether these factors contribute to the colony's large size. The case aligns with broader citizen science efforts like Map the Giants and related genomic testing initiatives that track giant coral colonies globally, even as taxonomy revisions for Pavona clavis continue. A new study from the Schumann Lab at the Max Planck Institute shows that stressed animal cells, including neurons, form inactive ribosome pairs called disomes through an RNA-RNA interaction, a regulated and reversible response to stress. The finding reframes how translation is controlled by RNA structure and expansion segments that regulate protein synthesis during stress, with possible implications for health and disease through ribosome organization and stress response. Researchers used a cross-disciplinary approach combining cell biology, biochemistry, yeast and mammalian genetics, and high-resolution imaging to manipulate ribosomal RNA and demonstrate the RNA-based mechanism behind ribosome oligomerization in stressed cells. Cryogenic electron tomography enabled direct visualization of ribosomes inside intact cells, revealing their reorganization under stress and supporting the disome model. The Science article, titled rRNA Expansion Segments Mediate the Oligomerization of Inactive Animal Ribosomes, was led by Aaron Schumann with key contributors Andre Schwartz and Mara Mueller. A specific ribosomal RNA expansion segment, 31B, mediates disome formation by forming a kissing loop interaction between identical RNA loops. Disrupting the 31B kissing loop prevents disome assembly, impairs cell growth, and increases stress sensitivity, indicating a regulated protective mechanism. The study shows how RNA structure and expansion segments contribute to translation control in animal cells, helping explain how cells conserve energy and recover quickly from stress. Overall, the work demonstrates a novel RNA structural mechanism of translation control and sheds light on ribosome organization during cellular stress. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.